In the scientific world, few mysteries have proven as persistent as the classification of the Tully Monster. Discovered in 1958, this 300-million-year-old fossil organism possesses a combination of features so bizarre that researchers have debated its classification for over 6 decades with no consensus, despite hundreds of specimens and multiple studies. In this video, we'll explore this prehistoric puzzle locked in time, examining the extraordinary anatomical features that make this creature so perplexing, from its stalked eyes to its pincer-tipped proboscis, and the conflicting evidence about whether it's a vertebrate like us or an invertebrate from a different evolutionary branch. What secrets might this ancient enigma reveal about how science confronts the truly unknown? The remarkable preservation of the Tully Monster is no accident of nature, but rather the result of a perfect storm of geological conditions at Mazon Creek. When these mysterious creatures perished in the ancient waters of Illinois, they weren't simply left to decompose like most organic matter. Instead, they were rapidly entombed in silty sediment, setting the stage for a remarkable chemical reaction. As bacteria began breaking down the remains, they released carbon dioxide, which upon meeting iron-rich groundwater, triggered the formation of protective siderite nodules around the decaying organisms. These natural sarcophagi effectively sealed the Tully Monster inside a time capsule, preserving delicate tissues that would normally vanish within days of death. This preservation process created an exceptional fossil record at Mazon Creek, capturing the actual soft tissue morphology of creatures from 300 million years ago. The siderite nodules formed three-dimensional fossils that included features normally lost to time, features that would later make classifying the Tully Monster deeply problematic. These ancient time capsules provided unprecedented clarity while presenting scientists with anatomical combinations that seemed to defy established categories of animal life. The scientific community first formally encountered this peculiar organism in 1966, when Field Museum paleontologist Eugene S. Richardson Jr. published his groundbreaking description in Science. Richardson faced a classification conundrum with a creature whose features didn't align with any known animal group. Despite having hundreds of specimens to study, Richardson established Tullimonstrum gregarium as its own distinct genus and species without assigning it to any existing phylum, acknowledging that this creature simply didn't fit into our understanding of animal evolution. The name gregarium, meaning common, reflected these creatures' abundance in the fossil record, making their absence from other sites worldwide puzzling. Richardson's approach implicitly acknowledged the questions that would drive scientific inquiry for generations. What was the Tully Monster? How did it live? Where does it belong on the tree of life? The creature's physical characteristics presented a classification nightmare. Its cigar-shaped body with segmentation suggested connections to invertebrate groups, while its triangular tail fin hinted at primitive vertebrate affinities. Most perplexing were those stalked eyes and that extraordinary tooth-lined proboscis, features with no clear evolutionary parallels. In 1989, Illinois designated Tullimonstrum gregarium as its state fossil, acknowledging both its scientific significance and uniqueness to the region. This bizarre organism had become part of Illinois' identity, connecting prehistoric life with modern communities. What makes the Tully Monster valuable to science is its bizarre anatomy and how it challenges our classification systems. When organisms don't fit established categories, we must re-examine fundamental assumptions about evolution. The Tully Monster reminds us that nature can present puzzles that resist easy solutions. Systems of classification falter when confronted with the Tully Monster's bizarre body plan. But what specific anatomical features make this creature so impossible to categorize? When paleontologists examine this enigmatic organism, they face a perfect storm of contradictory evidence. Its body simultaneously suggests relationships to several major animal groups while perfectly matching none of them. A truly perplexing evolutionary puzzle. The Tully Monster's most distinctive feature is its elongated proboscis, an anatomical structure with no direct modern equivalent. This flexible trunk-like appendage ends in a pincer-like mouth, equipped with up to 8 sharp teeth on each jaw. This feeding apparatus indicates an active predatory lifestyle, with the creature potentially probing muddy seabeds for prey or detritus. The unique configuration combines elements reminiscent of multiple phyla while matching none precisely. Equally confounding are the eyes, perched atop prominent stalks projecting perpendicularly from the body, a configuration without direct parallel in today's animal kingdom. The 2016 study in Nature by Clements and colleagues identified melanosomes, pigment-containing cellular structures within these eyes, initially strengthening the vertebrate hypothesis. However, other researchers challenged this interpretation, suggesting convergent evolution might produce similar structures in invertebrates. The presence of what appears to be a camera-like eye with preserved lenses further complicates analysis. Like a biological collage assembled from mismatched evolutionary parts, the Tully Monster combines features in ways that defy conventional classification boundaries. Its body represents nature's experimental workshop during the Carboniferous period. The classification debate largely centers on a structure interpreted by some as a notochord, the cartilaginous rod providing structural support in chordates. While this would place Tullimonstrum among vertebrates, its precise nature remains contested. Its characteristics don't perfectly align with typical chordate anatomy, and its relationship to other structures like potential gill pouches generates ongoing controversy. Some scientists identified what they interpreted as cartilaginous vertebral structures called arcualia, along with dorsal and asymmetric caudal fins, supporting the vertebrate hypothesis. Yet competing teams examining identical specimens reached entirely different conclusions. The complete absence of hard anatomical parts, no bone, chitin, or calcium carbonate structures, further complicates classification. This absence has led to proposals that the creature might represent a stem group to one of many worm phyla with few modern representatives. The Tully Monster exemplifies a phase of unprecedented evolutionary experimentation, where life forms tested configurations never again seen on Earth. Its unique anatomy suggests that 300 million years ago, boundaries between major animal groups may have been less distinct, with organisms exploring evolutionary possibilities later abandoned. The evolutionary experimentation of the Carboniferous period produced creatures that defy our modern classification systems. But in 2016, scientists believed they had finally cracked the Tully Monster's code. Their analysis of tiny pigment structures in its fossilized eyes sparked what would become one of the most contentious debates in modern paleontology. A scientific controversy that reveals how tenuous our understanding of ancient life forms truly is. When Thomas Clements and his colleagues published their groundbreaking study in Nature, the scientific community celebrated what appeared to be a definitive identification of the Tully Monster as a vertebrate related to lampreys. Their classification hinged on specialized pigment-containing organelles called melanosomes within the creature's eyes. Specifically, 2 distinct types, spheroidal and cylindrical, arranged in discrete layers, forming what appeared to be a retinal pigment epithelium, a structure characteristic of vertebrates. This discovery positioned the Tully Monster as potentially significant to understanding early vertebrate evolution. The arrangement of melanosomes seemed to place this bizarre creature on our branch of the evolutionary tree. Yet scientific consensus evaporated quickly. In 2017, Lawrence Salon's team issued a comprehensive rebuttal that systematically challenged the vertebrate hypothesis. They demonstrated that the presence of 2 melanosome types varies widely among vertebrates. Hagfish lack these melanosomes altogether, while some sharks possess only spheroid melanosomes. The supposed notochord inexplicably extended forward beyond the eyes. a feature never observed in vertebrates but seen in non-vertebrate lancelets. The rebuttal extended beyond eye structures. The body blocks identified as gill pouches didn't match lamprey anatomy, which requires complex skeletal structures absent in the Tully monster. Even the segmented appearance contradicted vertebrate anatomy, as true vertebrate myomeres extend continuously rather than appearing as distinct segments. Like a puzzle with pieces that refuse to fit, the Tully monster continued to confound classification efforts. In 2019, Rogers and colleagues discovered that certain squids and cuttlefish possess two different melanosome forms that can decay in patterns resembling vertebrate-like retinal layers. This revelation undermined a key piece of evidence for vertebrate classification. By 2023, advanced 3D scanning of 153 specimens led Tomoyuki Mikami's team to conclude that features previously used to justify vertebrate identity weren't comparable to vertebrate structures at all. Their analysis revealed body segmentation extending to the preoptic region, a pattern fundamentally incompatible with vertebrate anatomy, suggesting the creature might be a non-vertebrate chordate or even a protostome. When familiar classification criteria fail, researchers must rely on multiple independent lines of evidence. Yet the Tully monster continues to resist definitive placement, reminding us that nature doesn't always conform to our categorical boundaries. Nature's resistance to our categorical boundaries extends beyond the Tully monster itself to the extraordinary circumstances of its preservation. The Mazon Creek fossil beds represent one of paleontology's most remarkable paradoxes, a site where unique chemical conditions captured intricate details of soft-bodied organisms that normally vanish completely from the fossil record. Yet somehow this preservation miracle happened for an animal that exists nowhere else in the world. What makes this Illinois location exceptional is the remarkable preservation process. When organisms died in this ancient coastal delta environment, they were rapidly entombed in fine sediments, preventing oxygen from reaching the remains. At Mazon Creek, decomposing bodies triggered chemical reactions where bacteria produced carbon dioxide that combined with iron in the surrounding sediment, forming protective siderite nodules around the carcasses, nature's own treasure chests, preserving ancient life with extraordinary fidelity. The rapid burial and compression often cause carcasses to fold and bend, creating distinctive contortion patterns. These three-dimensional preservations stand in stark contrast to the flattened impressions typical of most fossil sites, allowing scientists to study anatomical details that would otherwise be lost. This preservation quality transforms Mazon Creek into scientific gold, essentially photographs from the Carboniferous rather than rough sketches. The ecological community captured within these nodules reveals the Tully monster's ancient world. It shared its tropical coastal home with a remarkably diverse assemblage of creatures. Sea anemones swayed with currents, primitive sharks navigated waters alongside horseshoe crabs, and early relatives of modern squids moved through complex food webs where the Tully monster played its mysterious role. This fossil site preserved an entire ecosystem in extraordinary detail, allowing paleontologists to reconstruct complex interactions between species. Yet one question remains particularly perplexing. Why is the Tully monster found exclusively at Mazon Creek? Despite numerous Carboniferous fossil sites worldwide with similar preservation conditions, not a single Tully monster specimen has ever been discovered elsewhere. This exclusivity suggests several possibilities. Perhaps the creature had an extremely limited geographic range. Alternatively, the Tully monster might have required precisely the right combination of environmental factors for preservation, factors uniquely present at Mazon Creek. The ancient environment-specific mixture of muddy estuaries and marine environments might have created ecological niches supporting this unusual creature. Thousands of unopened nodules from Mazon Creek remain in museum collections, each potentially containing new specimens or undiscovered species. Every crack of a nodule could reveal organisms with equally bizarre anatomies, possibly solving the Tully Monster mystery or presenting entirely new evolutionary puzzles. This single fossil site in Illinois represents a window into the past, a testament to how geological accidents preserve snapshots of life that would otherwise vanish forever. Beyond those unexamined nodules lies a curious paradox. Despite 6 decades of intense study, the Tully Monster continues to defy classification. What happens when paleontologists, frustrated by traditional approaches that yield contradictory results, turn to technologies that weren't even imagined when this fossil was first discovered? Enter synchrotron radiation, a technology peering into the ancient soul of the creature. These brilliant X-rays penetrate the siderite nodules, unveiling internal structures while leaving invaluable specimens intact. Where traditional methods risked destroying crucial evidence, synchrotron imaging preserves both fossil and context, revealing delicate anatomical features hidden within the nodule's stony embrace. Despite this technological arsenal, the Tully Monster guards its secrets fiercely. Mikami and colleagues' 2023 research demonstrates this persistent challenge. Their examination of 153 specimens using advanced 3D scanning techniques has dramatically undermined previous vertebrate classifications. Their detailed chemical analyses questioned anatomical features previously accepted as evidence of vertebrate identity. The alleged trilobed brain, tectal cartilages, and fin rays proved surprisingly dissimilar to their known vertebrate counterparts. At the microscopic level, scanning electron microscopy reveals cellular details, particularly regarding those peculiar eyes. By examining melanosomes, pigment-containing organelles, scientists compare these structures with modern animals. The pattern of these microscopic features provides vital clues about vertebrate or invertebrate affinities. Molecular paleontology brings another dimension to the investigation. Researchers extract and analyze organic compounds persisting within these ancient remains. Techniques like Raman spectroscopy identify molecular bonds in preserved material, potentially distinguishing between chordate tissues and materials like chitin. Biochemical evidence offering independent insight into the creature's true classification. Digital innovation joins the scientific chase. Computational phylogenetics and machine learning algorithms analyze vast data sets, comparing thousands of organisms against the Tully Monster's unusual anatomy. These tools test multiple classification hypotheses simultaneously, working to pinpoint the creature's position on the tree of life. Traditional paleontology remains vital. New fossils from other Carboniferous sites provide fresh context. Comparative anatomy, enhanced by advanced imaging, might reveal evolutionary relationships previously obscured by bizarre morphology. Each specimen offers another piece to this 300-million-year-old puzzle. This technological convergence on the Tully Monster mystery showcases how a single enigmatic organism drives scientific innovation. The classification quest has transformed into a testbed for cutting-edge research technologies and interdisciplinary collaboration, revolutionizing approaches to challenging fossils across paleontology. As these methods advance, we edge closer to answering the fundamental question: what exactly was this extraordinary creature that swam through ancient Illinois seas? The Tully Monster's enigmatic nature has pushed paleontologists to revolutionize their investigative approaches. Modern studies employing scanning electron microscopy, TOF SIMS, and Raman spectroscopy have transformed how we analyze fossilized remains, particularly for soft tissue examination. These technological innovations emerged from scientific necessity as researchers grappled with this biological puzzle. This single organism has catalyzed broader methodological advances throughout paleontology. Techniques refined through Tully research now illuminate other ancient mysteries. The classification challenge has enhanced our ability to reconstruct all forms of ancient life. The Tully Monster reminds us that scientific progress flourishes when confronted with nature's most perplexing puzzles. Each question bringing us closer to understanding our planet's remarkable past. Thanks for watching. We'd love to hear your feedback so we can keep making better videos. If you enjoyed it, hit that subscribe button on the end screen. See you next time.